Hi, I'm Ashley.
And I am Sharon, and we are here to talk about meiosis.
So Sharon, what is meiosis?
That is an interesting question. Let's find out. Meiosis is a process of cell division in order to make gametes.
And gametes are the reproductive cells found in the human body, which are the egg and sperm cells, right?
Yes, this type of cell is called a haploid, meaning that it carries only one copy of each chromosome. On the other hand, a diploid cell carries two copies of each chromosome, and these are the somatic cells in our bodies, such as the skin and liver cells. These diploid cells then undergo a different cell division process known as mitosis, but that is not what we're talking about today.
Then, can we talk about the cell cycle of meiosis?
Yes!
So, if I remember correctly, the cycle of a cell is divided into two main phases, interphase and M-phase. Interphase is when the resting cell is preparing to enter the division process, whilst M-phase is when the cell actually starts the cell division process, which is meiosis. Interphase has 3 stages and 2 checkpoints, which are the G1, S, and G2 phase, as well as the GO and G2 checkpoint.
That's correct. Now let's go further into interphase.
Well, a cell starts in the G1 stage of interphase and goes through growth, development, and differentiation. Now it is defined as a haploid cell, meaning that it carries chromosomes of 2N and 1X. Before entering the S stage, it has to face the G0 checkpoint, where the cell will decide whether it wants to continue the cell cycle and prepare to divide or lose its ability to divide.
Then if the cell decides to continue to divide, it will then pass to the S stage where it will replicate its DNA in a process called DNA synthesis. After the cell copies its DNA, it will become a diploid cell, thus it carries 2 sets of chromosomes. The chromatids are then joined together at the centromere. After the cell duplicates its DNA, it enters the G2 stage and prepares for cell division.
Okay, then, during this stage, the cell will replicate organelles, store food, and make protein. When the cell is finally prepared to divide, it will face the G2 checkpoint to verify if the cell's DNA is damaged or not. If the cell has undamaged DNA, it will enter the M phase. However, if the cell does not have damaged DNA that cannot enter the M phase, it will have to fix the damaged DNA or it will be sent to apoptosis, otherwise known as programmed cell death. Have we finished the cycle now?
No, this is when meiosis starts.
Oh, okay, on with it then.
The cell is finally ready to continue its journey into the M phase, where it will go into meiosis. When a cell goes through this process, it will have to go through it twice, thus obtaining the name meiosis 1 and meiosis 2. Both of these share the same process: prophase, metaphase, anaphase, telophase, and cytokinesis.
Okay, so in prophase 1, which is during meiosis 1, the chromosomes condense and they become visible. These will then find their homologous pair and will stick together, forming a tetrad. Within the tetrad, the chiasma of the chromosomes will exchange parts of the DNA, resulting in crossing over. Then, the centrioles and spindle fibers around the centrosomes will begin to form. The cell's nucleus will envelop and will also break down. And then chromosomes will begin to move towards the central line of the cell, called the metaphase plate. The centrosomes will also begin to move to the opposite poles of the cell.
Yes, when the tetrad lines up in the middle of the cell, the spindle fibers formed during the prophase will attach to the tetrad at the kinetochore of the centromere. This is when metaphase 1 begins, where each duplicated pair will be attached to a microtubule. At this point, the nuclear envelope has disappeared, and then the centrosomes at the poles will begin to separate the pairs of homologous chromosomes towards the centrosome. And as the kinetochore microtubules start to pull the attached chromosomes towards the ends, the cell will consequently elongate.
Okay, so if I'm right, the duplicated chromosomes will reach the ends of the cell and the nuclear envelope will start to form again around the chromosomes. Then, during cytokinesis, a ring of microfilaments will create a cleavage furrow to divide the cytoplasm, and the cleavage furrow will pinch between the two new nuclei. Ergo, two new cells will be formed.
Yep, that's how it works. So now let's go to meiosis 2.
Wait, there's a part 2?
Yes, Ashley, now pay attention. So first of all, meiosis 2 is similar to meiosis 1, but instead of separating the tetrad, the homologous chromosomes are separated, which were the end of meiosis 1. At this point, each cell would have one of each chromosome, thus 1 and 2x.
In prophase 2, the new cell's nuclear envelopes break down again. The meiotic spindles begin to form where they attach themselves to the chromosomes, centromeres. The chromosomes subsequently condense and move towards the metaphase plate.
Yes, and metaphase is when the chromosomes arrive at the center of the cell, or the metaphase plate, and all the kinetochores are attached to the spindle fibers.
This change is due to the fact that the chromatids are being separated, and as this process continues, the cells will also elongate. When the chromosomes have finally arrived at the center of the cell, at the metaphase plate, and all the kinetochores are attached to the spindle fibers, then that's when the chromosomes are starting to separate and move towards the end of the cell. The number of chromosomes will change from 1n, 2x to 1n, 1x.
At this point, each nucleus will contain only one set of chromosomes with only one chromatid. Following that, the nucleus fully forms and surrounds the chromosomes.
In cytokinesis, a ring of actin is formed from microfilaments between the two nuclei. In this process, the cytoplasm divides and starts to divide the cytoplasm, and the chromosomes uncoil.
Now, meiosis 2 has finally finished, and 4 cells that carry different DNA are formed.
Wow, that was so cool.
Another topic related to meiosis is spermatogenesis and oogenesis. Oogenesis is a development of ovums, and spermatogenesis is a development of sperms. In each development of the gametes, meiosis occurs, but these two developments are not exactly the same.